Thank you so much, and welcome back from the break. Let me just tell you what an honor it is to be attending this conference with many of my heroes. So there's a lot of people in the room here who have influenced me, and I have to tell a particular story. I study social networks, and one of the things that I've worked on with Nicholas Christakis, my longtime collaborator at Yale, is looking at indirect effects, person-to-person effects. And my good friend in graduate school is Eric Dixon, who brought back from some time with Ken Binmore a number of ideas about how you could merge the social sciences and the biological sciences, and that's been deeply influential to me. So it's a real honor to be here. Today I want to talk to you in particular about friendship and natural selection. Now, you don't always hear a social scientist talking about evolution. It's because we often think about these processes from a first-person point of view. We think about friendship as something that we as individuals engage in. But in fact, as a species, friendship is something that makes human beings unique. There's no other species that connects to people that we're not related to and that we're not interested in reproduction with on such a large scale as human beings. You look at the other social species like ants, for example, they're all related to one another. You look at other non-human primates, those groups are primarily made up of relatives. It's unusual for there to be interactions between non-related individuals. And so friendship is something, if you want to understand humans, you really have to understand friendship. But in order to be able to understand this from a scientific point of view, we want to be able to make some kind of connection between what we commonly think of as friends and how we would study this scientifically. So let me ask you guys to engage in this process and think about your own networks. I want you to answer this question for yourself. Who are your friends? This is not optional. I want you to make a list in your head. First name, last name. Who are your friends? Now, you guys are all good scientists, so you might be thinking, well, maybe we don't know what the definition is. Maybe this is like trying to define fairness. And so let me give you a little bit of help. This is usually what we do to prompt individuals when we were asking people to give us their friendship networks. We ask them to name these kinds of people. Who do you discuss important matters with? And who do you spend your free time with? Now, you notice that those might not be overlapping sets of people. You might have people at work that you spend time with, but you don't really discuss anything important with. Vice versa, you might discuss important matters with these people, but outside of work, you don't actually see them at all. And so one of the things that's important to realize is that there's different kinds of ties, but a lot of this has to do really with the amount of time that we're spending with people and the quality of those interactions. So are you done? Do you have your lists, first name, last name? Okay, so now what I want you to do is I want you to think about, suppose that we did this all around the country with Gallup, which we did. What average do you think we would find in the number of these deep, close, real-world social connections? Well, as it turns out, when we asked people to do this exercise all around the country, people named between 0 and 8 people and averaged about 5. So think about your own network now and think about where that puts you. Because those people that you named aren't islands. Probably they also have friends, and they're also connected to other people, which means they live in networks that might look like this. And the very first way that you guys are probably different from one another is that some of you have more friends than other people. Okay, so for example, we have 2 different people in this particular network where each circle is a person, each line is a relationship. And you have person B and person D who are both on the edge of the network, but one of them has 4 friends and one of them has 6 friends. Okay, so that might describe the 2 of you. But do you know where you might sit in this network? You kind of need the whole map to be aware of where you sit in this network because all you see are those direct connections. Now, from the point of view of evolution, you can imagine that there are reasons why we might want to have more or less friends. Friends might mean social support, right, for example. And so the more people that you're directly connected to, the more people might be there, for example, to share blood with you if you're a vampire bat, or to give you other kinds of resources in the ancestral environment to help you survive. But what about these indirect connections? Well, for example, you might have the same number of friends as somebody else. So person C and person D, for example, both have 6 friends. But person C has many more friends of friends, which puts them closer at the center of this network and a smaller number of steps from everybody else in the network. Now, do you want to be person C or do you want to be person D in the ancestral environment? Well, suppose there's a disease that hits the population. Or as another way I put this, if the Black Plague is rolling through Europe, do you want to be the life of the party or do you want to be a hermit? Okay? You probably want to be on the edge. You don't want to be that person in the center. Diseases will wipe out the center depending on how virulent they are. Right? On the other hand, you can imagine that there's pieces of information flowing through this network that are very valuable, in which case you want that information and you might actually want to be that person at the center. So you can imagine a series of changing environments over time that would enforce this kind of diversity in both the number of friends we have and the number of friends we have. And there's another property of human networks that makes them unique from other kinds of networks, and that's transitivity. So consider person A and person B. They both have 4 friends, but none of person B's 4 friends are connected to one another. Whereas person A, most of person A's friends are connected to one another. A lives in this tight, dense group where everybody knows everybody else. They're all buddies. They all go out and do everything together. Whereas person B has a friend in that group, a friend in that group, and a friend in that group. So think about, again, in the ancestral environment, which one of those 2 things would have given those individuals more fitness? Okay, one possibility is you want to be person A, because person A is going to have people watch his back. If he wants to bring down a mastodon, those guys are going to be there for him and they're going to protect him in the pursuit of that mastodon. But if you want to find the mastodon, if you want to get different points of view, more information, person A is not going to be the best location in the network, because he'll turn to his buddy and say, do you know where the mastodon is? No, I know the same thing Harry does. Do you know where the mastodon is? No, do you know the same thing? They all have the same information, whereas person B has access to different information. So if you want to find the mastodon, you should be person B. And depending on the environment, depending on whether or not information is more important or social support is more important, you might get variation over time in what kinds of structures would be the ones that we would expect to emerge from an evolutionary process. And so perhaps that's the reason why we don't look like this, why we all don't have exactly 4 friends or 8 friends in a lattice. Why instead there's variation in the structure because of the variation in the environments that would have enacted selective pressures on us. And maybe those distributions, maybe there are regular principles that govern those distributions because human social networks tend to have consistent structure. And so just for example, we have teenagers in the upper left and college students in the upper right, and people like you and me, academic collaborations here in the lower left, and people who call one another on the telephone in the lower right. These networks tend to exhibit similar properties in the distribution of the number of friends, for example, in the distribution of the path links from one person to another in the network, in the tendency to engage in reciprocal ties, in the tendency for us to close triangles, to introduce our friends to one another. And this is something that's really motivating Nicholas Christakis and I right now to think about, where do these structures come from? Is it possible that evolution is involved, and to what extent might genetics actually be involved? Another important structure that's very characteristic of human networks is the tendency for us to be with similar others. And so in this work with Nicholas, for example, from the Framingham Heart study, we mapped the social network of individuals in this long-running study where each circle is a person, each line is a relationship, and we put information about the people in the circles themselves. In this case, we were studying obesity, and we made the circles larger for larger people. It's proportional to body mass index. And what we discovered is that there are clusters in this network that extend beyond our social horizon. They extend to our friends' friends and to our friends' friends' friends. In fact, we can characterize this statistically. If I don't know anything about you, but I randomly poll one of your friends and you're a member of this study, if that person is obese, it increases the likelihood that you're obese by about 40%. If I poll one of your friends' friends and they're obese, it increases the likelihood that you're obese by about 20%. And if I poll one of your friends' friends' friends, it increases the likelihood that you're obese by about 10%. And beyond 3 degrees of separation, we have not found those kinds of relationships in the Framingham Heart study. They tend to go out to 3 degrees of separation and no further. And that's true not just for obesity, it's true for smoking behavior, drinking behavior, happiness, loneliness, depression. Divorce clusters extend out to 2 degrees of separation to your friend's friends. If your friend is divorced, you're more likely to be divorced yourself. People at this point, after all these studies, were starting to say things like, well, are these Framingham people just a little weird? Maybe you can go and get some other data to see whether or not this replicates in other places. And so we went to the National Longitudinal Study of Adolescent Health and replicated the obesity finding. And we also were able to show that the use of marijuana and sleep loss are also correlated in clusters in this network. And we didn't find exactly the same thing that we found in Framingham. Instead of these clusters extending out to 3 degrees of separation, we found that they extend out to 4 degrees of separation. If I don't know anything about you, but I know that your friend's friend does marijuana, then statistically I should guess that you do marijuana as well. And so these clusters are a regular feature of human society. And we've been very interested in also looking at how these might be enacted online, because we now have these wonderful, rich data sources of connections online, many of which are proxy for real life. But the problem is that there's not necessarily a direct correspondence between the online world and the offline world. So take, for example, this is a close dorm at Harvard, and we ask people to give us their lists, like you have your lists in your head. You can imagine if this was your college dorm, where would you have been in this network? And now we also ask these people, well, give us your Facebook networks, and look at those interactions, and that's what those look like. It's a big ball of spaghetti. The average number of connections on Facebook is 150. And so people are connected not only to their friends, to their friends' friends, and their friends' friends, and to strangers. And in fact, when we try to find the same kinds of clusters online, we can't find them. They don't exist. There's nothing about your average Facebook friend that is predictive of your own behavior. So what does this mean? Where does this disconnect come from? It comes from the fact that the strength of relationship is so weak that you share nothing in common with these people. But that doesn't mean that you can't use this information to get at those close friendships. And the way we did this originally was by using information like photo tagging. Okay, so the average person has 150 friends, but they have, on average, 10 photo friends. These are people that they've uploaded photos of and tagged. So for example, this is a picture of my lab and me at the movie The Social Network, where I tagged all of the people, and we're all connected on Facebook. It's very meta. But, so the idea here is that you can actually use information like this to recover the real-world social network. And when we do that, we get networks that look more like Framingham again. Networks look like this, where each line now is a photo-friend relationship, and you start to see patterns of behavior. And we started with patterns of behavior that weren't necessarily so relevant. So for example, this is the a map that shows you whether or not you're smiling in your profile picture or not smiling in your profile picture. And here we get clusters that extend out to 2 degrees of separation. That's Nicholas. I always have to make fun of him. But we also found that this was true for other behaviors that were more important, like how much you weigh. And we found clusters of overweight women and clusters of overweight men that extend out to 2 degrees of separation. So this is a fundamental property. We tend to live in neighborhoods in our social space where people are more or less like us. But why? One possibility is that this is just a false positive, right? So that you can just throw a bunch of 1s and 0s at a network, and you're going to get clusters of 1. Sometimes you flip 6 coins, and you get 6 heads. That's actually a relatively easy problem to solve, because you can just permute the network. You can see how likely the observed correlations are to come about due to chance. Another possibility is due to selection effects. And here we're talking about evolution. Selection is really important. Not just natural selection, but human selection. We choose our friends. And sometimes we choose our friends based on the characteristics that they convey. So normal weight people, for example, might choose to become friends with other normal weight people. Another thing that can influence us to be similar is contextual effects. There might be things in our environment that will cause us to become similar. So a McDonald's opens up down the street, and you and your friends both independently start chugging Big Macs, your weight will go up, and it'll create one of these clusters in the network. And so correlation in environmental stimuli is another thing that can generate these clusters in human social network. Now, the factor that we have been most closely studying is influence, which is this idea that not only do we choose our friends, but we shape them, and they shape us. And so if I start losing weight, if I go on a great diet, and I feel good about myself, and I share that information with my friend, they may be willing to do the same thing. Or maybe they're a carrier of that disease, and they don't change their behavior, but they tell their friend, my friend's friend, about this diet, and now they start dieting, which helps to explain why these 2-degree effects and these 3-degree effects might matter in these closed neighborhoods. This is what we've primarily been focused on, but in these observational studies, it's very hard to distinguish these different kinds of effects. There's currently a really robust debate in statistics about the extent to which you can do such a thing. So what we have done recently is we've actually gone to experiments. So for example, we did experiments with public good games. And this is a paper where we analyzed somebody else's experiments, but we did related experiments in our own labs. And what we did is we had people play in groups of 4 public goods games. And in a public goods game, you come in, and I'll let you think about what you would do if you're in this experiment. You come in, I put you in a group of 4 people, I give you $10. I give each of the other 3 people $10 as well. You decide how much of that money to keep for yourself and how much to give to the group. All of the money that everyone gives to the group, I, the researcher, am going to double, and I'm going to redistribute it equally. You might say fairly, or you might not, actually, given this morning's conversation. But basically, we're going to be dividing it equally. So think about what you would do in this case. Well, if you're a self-interested profit maximizer, every dollar you give, you're only going to see 50 cents of it come back to you, right? Because it gets doubled and split up 4 ways. So if the world is full of self-interested profit maximizers, nobody ever gives anything in this particular experiment. But in fact, behavioral economists have shown this in spades at this point, that people do give some things in this experiment, and they tend to be influenced in their play based on what other people do. Now, some of that has been explained based on reciprocity. So I'm going to do to you what you did to me in the last round. But in this case, we randomly assigned people to be in different groups at every round. So they never played with the same person twice. So the question is, how much does my behavior affect you when you play with different people? And what we found in this game is that every extra dollar of giving in the public goods game in the first round caused people, when they played with other people, to give an extra 20 cents in the next round. And those people who played with them gave an extra 8 cents in the next round. And those people who played with them gave an extra 5 cents in the next round. In other words, through random assignment, we were able to show that there's some influence effect here that extends beyond our social horizon, that at least some of these clusters can be explained by influence. But you still have to think about evolution. Would evolution have favored this kind of process, where we would coordinate our behaviors in this kind of way, where we would come to cooperate with one another, where we would come to copy the other people in our group? And we've shown that these influence effects exist not just in the laboratory, but also in real life. And in a paper we published last year, we discussed an experiment we ran in 2010 in the US congressional election, where a single message on election day got an extra 60,000 people to vote. And this message that we posted on Facebook. We randomly assigned it to people. And the reason why we did this is because we wanted to see whether or not the friends of the people who received the message were also themselves more likely to vote. And we found that that got an extra 280,000 people to vote. Not seeing the message themselves, but having a friend see the message. And self-reported voting, clicking on this I voted button, that also spread beyond people's social horizon. Your close friend's close friend was more likely to click on the button if you clicked on the button. And so what we're starting to see is that in these online settings where we try to recover these real-world networks, we're still finding that some of this is an influence effect. And so basically what we found is that there's a number of these different kinds of attributes that we share in common with people that are socially connected to us. And we started to wonder, why might this be the case? Why is it the case that it's about 3 degrees plus or minus 1? What is it about that measure that might be consistent across these different populations and these different phenomena? And so we started looking at works like this work by Robin Dunbar. Because we thought that maybe there's something about the number of people that we reach with this process. And if you think about the average person in America having 5 close friends, abstracting away from overlapping connections, that means you have about 25 friends of friends, about 125 friends of friends of friends. Okay? So we're talking about order 1 to 200 people that will be indirectly touched by something that we do. Well, as it turns out, this is also very close to natural human group size, according to Robin Dunbar, where what he did to estimate natural human group size is he compared the neocortex ratio, the part of the brain that's devoted to the neocortex, to mean group size in a bunch of non-human primates. And then he took our brain size and extrapolated to get 150 as the number for what we must have evolved to cope with in terms of our natural groups in the ancestral environment. And so maybe this is one of the reasons why we influence people out to 3 degrees and why we're influenced by other people out to 3 degrees. It's because of that coordination where we can get groups to do things together, or we can cooperate with groups out to that limit in order to be able to achieve better fitness in that environment. Now, we weren't really sure whether or not that was right. And so one of the questions that we had is that if this is true, then we ought to expect that social networks in the ancestral environment look a lot like our ancestral networks today. And so in order to test this, we actually went to the Hadza. It's a hunter-gatherer group in Tanzania, Corina Pachela. She went there with a photographic census that was created by Frank Marlowe, who studied the Hadza for a long time. And we showed them the census, and we asked them to give us their friends, the same way you guys have done. So they did more or less the same thing that you guys did. We used a different name generator. We asked, after this camp ends, with whom would you like to camp? We also gave them honey sticks and asked them to give the honey stick to anyone that they chose. We mapped those two different kinds of networks. And what we found in those networks is that their networks are like ours on each one of these different kinds of characteristics. Degree distribution. Most people have a small number of friends, and a few people are super connectors. They have a very large number of friends. Homophily. They tend to be clustered by age. They tend to be clustered by gender. They tend to be clustered by food preferences. There's on a wide range of different categories, they tend to be clustered with people that are similar to them. Degree assortativity. The more popular Hadza tend to be connected to the more popular Hadza. And this is really interesting because this is something that we don't find in non-human networks. The internet, for example, if you're Google, you're connected to a bunch of very, very low connected networks. There tends to be negative assortativity in these other kinds of networks compared to human networks. Geographic distance. We tend to make friends with people who are near us. Relatedness. We tend to make friends with people who are genetically related to us. Reciprocity. If I named you as a friend, you are very, very likely to name me as a friend back. There's a low asymmetry in those relationships. And then transitivity. This is something that a lot of our theoretical models of social networks don't do a good job of generating. These triangles where not only are you connected to each of your friends, but your friends are connected to one another. Like that person A in the lower right-hand corner, we tend to live in cliques, in groups where people are connected to one another. So on each one of these, we found that the Hadza's networks are like ours. We also had these Hadza play public goods games and looked at their levels of donation. And so, for example, here's the network of relationships. So there's the different kinds of relationships that we mapped. And the blue nodes are high donations, greens are medium, and yellows are low. And you can see there are clusters of blue and clusters of yellow. And we mapped these separately for males and females. In fact, one of the things we were able to show is that within camps, you have low variance, lower than expected variance in cooperative behavior. There are some camps that cooperate more and some camps that cooperate less. And that between camp variance is actually higher than you would expect. Well, as it turns out, these are exactly the conditions that, according to the price equations, you would need in order for altruism to be able to survive. And it doesn't matter where it comes from. It doesn't matter if I'm influencing you to change. It doesn't matter if we choose people who are like us. The fact that you have that local cooperation is something that can protect people who are altruistic in these local groups because those groups tend to do better than other groups over time. And it's not that selection is acting on the group, it's acting on the individual, but because of the difference in variance, the individual's behavior towards cooperation tends to propagate over time. And we found this not just true between camps, we found this true in the network itself, where we found that, that there was clustering in cooperative behavior that extended out to 3 degrees of separation. So we started thinking from this then, well, maybe there's something that genetics might help to explain in terms of all this. Because if we have the same kinds of networks now that we had 30,000, 100,000 years ago, then maybe there's something that's really stable that's helping to form these networks. And what we realized is that there's already a wide range of studies on genetics influencing a lot of the very same phenomena that we have been studying. So for example, it appears that genes have an impact on obesity. It appears that genes have an impact on smoking. It appears that genes have an impact on happiness. It appears that genes have an impact on cooperative behavior. And so the idea here is that we had been studying social networks and their impact on behaviors. A number of other scholars had been studying the impact of genes on behaviors. And we started wondering if there's sort of a general purpose mechanism that helps to explain some of this relationship via social networks. In other words, maybe selection was acting in general on our tendency to form these relationships and the patterns with which we would form them, which then has implications for the kinds of behaviors that we would exhibit both as individuals and in groups. And so we started doing some studies like this one, where we wanted, first of all, to understand the structure of these social networks and how they're related to genes. And in this case, what we did is we did a classical twin study where we compared fraternal twins who share 50% of their genes on average to identical twins, which are essentially clones. There's some copy number variation and things like that, but if you genotype them, they're going to have the same allele at every one of the markers that you find to a very, very small degree of error. And what you would expect to find if genes were playing a role in social networks is that the identical twins would have more similar social networks than the fraternal twins. And that's what we found. And we actually used the classical twin method to estimate heritability in this case. And what we found was that in degree is heritable. So the number of people who name you as a friend is heritable. Okay, so the number of people whose list you are on. And if you think about that, there's some nice mechanisms that might cause that, right? So we know that physical attractiveness is one of the things that increases the number of friendship nominations. And physical attractiveness is a function of morphology. I don't think anyone in the room is going to say that morphology is not at least a part of and partly explained by our genes. But we also found that transitivity is heritable. So transitivity, the probability that two of your friends are friends with one another. I find this truly extraordinary. What this means is that your genes are explaining some of the variation in whether two of your friends are friends with one another. So it's not just you and your genes, it's you and your friends' genes. It's not just the genes in the environment, it's the genes and your friends' genes as the environment, or the environment having an impact on the kinds of friends we choose and the kinds of genes that we're exposed to. All of a sudden, this dramatically complicates our picture, this individualistic picture of one set of genes and one person. And so after this particular study, where we were looking at the effect of genes on, potential effect of genes on structure, we also wanted to try and see whether or not there was homophily in genotypes. And so the idea here is that if genotypes can help to explain certain phenotypes, and if we're sorting based on those phenotypes, then we ought to also expect that there would be assortment on certain genotypes. And in fact, it might be easier to detect this for complex social behavior, because there may be many genes involved in a complex social behavior, and many genes involved in somebody else with a complex social behavior, where you would expect genetic similarity. It would be hard to find any one of them, but the set of them might be easy to find because you have many ways to measure them. In this particular case, we had candidate gene studies available to us in the National Longitudinal Study of Adolescent Health, and what we found was that there was clustering in this particular gene, positive clustering. So if I don't know anything about you, but I know that you are a member of this study, and I know that your friend has a certain genotype, a certain variant of this DRD2 genotype, then I can do better than chance in predicting whether or not you have this genotype as well. And that's net of ancestry. It's net of control for ancestry that we did in this particular population. We replicated this in the Framingham Heart study, where we had a much better control of ancestry, the first 10 principal components of the gene subject matrix for an anthropometric strip with 500,000 people. We also replicated a negative correlation in CYP2A6, where if I don't know anything about you, but that you have this genotype, then I can predict that your friend has the opposite genotype. And so this was just the beginning, though, of thinking about this, because we all know the problems that candidate gene studies have had over the last 10 years, right? And it's hard to know whether or not this will survive multiple replication attempts. It's survived these two things, but given our past history and given the low effect size, it's possible that these are false positives. And so one of the things that we started thinking about is well, what are some other things that we could possibly say that wouldn't necessarily rely on making statements about single markers? And so we developed a theory about what kinds of markers might come about. And this is based on this overall question: why do we choose similar others as friends? Why do other species choose similar others to engage with? Why is homophily so ubiquitous? Why isn't it equally plausible that we reach out for individuals that were different than us? And especially if you think about the benefits of specialization. If you're an economist, you probably are thinking, like, there's lots of things that I could give to another person, and they could give me something in exchange. Right? So why don't we see that more often than we see a situation where I'm producing exactly the same thing as this other person is producing, and we tend to get together. So we created this very, very simple evolutionary game theory model with Martin Nowak, where basically you interact in a population. If you successfully interact with another individual in this population, if you interact with someone who's the same phenotype as you, you're going to get a payoff of A. If you interact with someone who's the opposite phenotype of you, you're going to get a payoff of B. But whether or not you interact is going to depend on whether or not you want to interact with someone who's a similar type or someone who's a different type. So we let natural selection act not only on the evolution of phenotypes, but also on the tendency to choose similar partners. So we have this parameter P, where 0 is the tendency never to choose a similar type interaction, and 1 is to always choose a similar type interaction. And vice versa, 0 means you always choose the opposite type interaction, and 1 means you never choose it. And what we found was that under certain circumstances, you would expect homophily to evolve. And so this is the limiting distribution of this parameter p in the population. This is stochastic, so this is the reason why there's some noise. And so you would tend to favor homophily in some populations. You would tend to have both heterophily and homophily favored in some populations. And in some, you would have heterophily favored in certain populations. But what was really interesting is that this parameter A, that tells you how much the fitness advantage there is to same type interaction, it can actually be lower than B, and you still get homophily. The advantage to homophily can be lower than the advantage to heterophily, and you still get populations that tend towards homophily. Now, why is that the case? It's because it's easier to find a successful partner if you're looking for a same type person in a monomorphic population than it is to find a partner with whom you're going to be successful in a polymorphic population. There are fewer of those individuals. There's more of a chance that you're going to mess up. There's a higher cost to developing a successful relationship in those kinds of populations. And so we would expect homophily actually to be ubiquitous, and we would expect these kinds of relationships with the tendency for those different phenotypes under natural selection to exhibit different kinds of fitness. And so what we did now is, in order to be able to kind of test this theory, is we went back to the Framingham Heart Study data, where we have about 500,000 unimputed SNPs, and we can use technology to impute SNPs. We used Mach to impute about 1.5 million of these SNPs, and we did this for about 2,000 subjects and about 1,300 pairs of friends in this relationship, because what we want to know is whether or not these individuals tend to gravitate towards other people that are genetically similar to them. We can use as a comparison group about 1 million stranger pairs in this data set, where what we do is we match people that are not relatives and they're not friends. And it's great because the Framingham Heart Study has really good detailed information on family structure and also on friendship, so that we can, basically if you don't have a connection, there's a very strong likelihood that you don't have a connection in real life. And we can look at the set of all of those, and since networks are so sparse, the chances are very good that any kind of contamination from someone that you actually know that's not documented would be very low. Well, the first thing that we find is that when we measure the kinship coefficient in these friendship pairs, that it's shifted to the right relative to strangers. What do I mean by that? Kinship, if you multiply kinship by the kinship coefficient times 2, you get relatedness, essentially. And relatedness, you probably have heard this before, right? 0.5 relatedness is you and your sibling, or you and your parent. 0.25 is you and your grandparents. And so kinship, what this is telling you is the probability that you're going to have the same alleles at the same locus across the whole genome. And what we find is that compared to the strangers group, that friends tend to be more similar. And this is after removing all of the people that have an explicit relationship and removing anyone who had a kinship of greater than 0.01. Now, this result does not control for population stratification, but the strangers are drawn from the very same population. So at least we know that this isn't a consequence of cryptic relatedness. But still, it's a possibility that this could be due to, for example, someone becoming friends with a third cousin that they actually know about, or maybe they don't know about it, but there are family relations that sort of brought those two people together. So ideally, what we would like to do is we'd like to do something a little bit more strict. And so we do a genome-wide association study, where now at each locus, rather than looking to see whether or not a behavioral phenotype is associated with a genotype, we look to see whether or not the friends, or whether or not your own genotype is associated with your friend's genotype. And what we do in order to control for ancestry is we actually include the first 10 principal components of the gene subject matrix, both for you and for the friend. We focus on common alleles only, because there's the problem here is that if your genotype is rare, then your friend's genotype is also rare. And so a lot of studies, they'll restrict to minor allele frequency of 0.01, for example. But the problem there is that you get inflation in the statistics if both of those things happen to be rare. So we make sure that they're sufficiently common. We also use multi-way clustering to account for repeated observations. And crucially, what we're going to do is we're going to do the exact same GWAS in a set of stranger pairs that have exactly the same network structure, but they're connected to people that have no relationship to and they're not friends with. And when we do that, first of all, what we find is that the friends actually exhibit more inflation than the strangers, significantly more inflation than the strangers. Now, some people might be worried about inflation in the strangers here, 1.02, it's not high, but it's not low either. It's sort of moderate. But the idea here is that the friends are even more inflated than the strangers, and there's a nice paper that just came out last year that suggests that this can be due to the fact that the trait that you're observing is polygenic. And we would not expect homophily to be exhibited on just a single locus. It should be exhibited on many, many locuses if complex social behavior is involved. We also are able to show that it's not just the case that you have this excess number of p-values for the friends relative to the strangers, but they're happening both for heterophilic SNPs where we tend to be different from our friends on, and also homophilic SNPs, friends that we tend to be the same on. And both of those are significantly enriched relative to the strangers. Just to give you an idea of what these Manhattan plots look like, this is showing that we actually have even some hits that survive genome-wide corrections for multiple testing. In this case, it's a Bonferroni correction, so it's very conservative. But we do not make any claims about those SNPs. I do not think that those particular SNPs would necessarily replicate in another sample because we only have 2,000 people in our sample. And although the friends actually exhibit patterns, you can see these sort of the patterns of excess relationship here compared to the strangers. What we really want to do is, rather than focusing on individual genes or individual SNPs, what we want to do is we want to focus on sets of genes, because that's where I think we have some chance of being able to identify enriched relationships that would replicate. We also want to be able to try and see whether or not we can use the GWAS to predict out-of-sample behavior. So once I have all of these weights, a lot of those weights are going to be nonsense, right? There's going to be a bunch of false positives in those weights, but there's going to be some information in them. And so maybe I can use those weights that I get from the GWAS then to go and genotype 2 of you in this room and then make a prediction about whether or not you're friends. And so the way we did that is we held back a set of people who were friends and a set of people who were strangers. And we used this model that we fit on another set of data to predict whether or not they were friends. And as it turns out, it does significantly. So the one standard deviation increase in this friendship score that we created increases by 6% the likelihood that you're friends with this other person, and explains 1.4% of the variance. Now, I say this very triumphantly, and for people who aren't engaged in genetics research, you might think 1.4%, that's ridiculous, that's nothing. But you have to take this into context. Even things we've studied to death, that we've got literally billions of dollars pouring into, our best models are explaining 0.4 to 3.2% for schizophrenia and bipolar disorder, and body mass index about 1.5% in these out-of-sample tests. So this is just the beginning. I think if we had a much larger sample, we'd get more precision, and we would actually get more prediction out of sample. And also, this is an additive model, right? So it doesn't account for interactions. It doesn't account for any gene-environment interactions. There's a lot of things that you can imagine that would eventually improve the fit of the model. When we did ontology analysis, the strangers' GWAS produced nothing. That's good. You wouldn't want it to produce anything. When we did the friends analysis, we found 3 gene sets that actually mapped to both homophily and heterophily. So olfactory transduction is one of the sets that looks like it's associated with these homophilic SNPs. In other words, it looks like we tend to make friends with people who genetically share the same sense of smell that we do. And this was previously hypothesized when we were trying to think about mechanisms that might generate this kind of homophily in networks. And in fact, this is one of the things that has been proposed for heterogamy as a mechanism. And so there's some evidence, although it's contested, that we tend to choose spouses, for example, who have different HLA genotypes from us in order to be able to make sure that we're surrounded by someone who can defend against diseases that we're susceptible to. And there's another set of papers, also contested, which suggests that one of the ways that we select those individuals is through a sense of smell. It's not clear what the endpoint of that literature is going to be, but it's another sort of parallel area where it looks like the olfactory system might be important to the mechanism. Another thing that we find is the linoleic acid metabolism. We have no idea what that means. Possibly it's a false positive. But we find that heterophily actually has this relationship with the immune system process, which has an analog in these results from the literature on heterogamy. And just so you can see, another thing we checked was whether or not the network within the genes for the olfactory gene network, if we were enriched, say, in the center of that network. And it doesn't look like there's any patterns there. But maybe we'll find something whenever we can ensure that there are fewer false positives in the specific associations with specific genes. And so the last sort of slide that we wanted to get to here is one where we wanted to ask the question, are these SNPs that are more homophilic, are these SNPs that we share in common with our friends, are they the ones that are under the most recent rapid natural selection? So we took Pardis Zabedi's common, or her score of natural selection, and we compared it to genotypic correlation, where we have the heterophilic SNPs on the left and the homophilic on the right. We did this for the strangers and got no result, but for the friends, what we found was that the genes we share in common with our neighbors are the ones that are more likely to be under recent rapid natural selection in the last 30,000 years. And so it's possible that this tendency for us to assort with people who are like us might be speeding up evolution, at least at those points and possibly at other points along the genome. And in future work we've got going right now, we've looked at spouses, both in the Framingham Heart Study and in the Health and Retirement Survey. And here what we find is the same result for spouses. So spouses tend to choose other spouses, and those genes that are similar tend to exhibit this property, but also the genes that are dissimilar. So in our theoretical paper, there was that intermediate case where you would expect natural selection to be favoring both heterogamy and homogamy, and that's what we appear to be finding here whenever you have multiple phenotypes. And we've done simulations to show that this can help to explain some of the speed up in evolution that's happened in human behavior over the last 30 years. So let me just conclude by saying what I think this research is tentatively suggesting is that we are metagenomic, not just with respect to the organisms within, like this fine paper was able to demonstrate, that we are the product not just of our own genes, but the genes of all the organisms that live within us. We are also metagenomic with respect to other people outside of us. And I think it will be interesting for us to think about to what extent are our friends' genes a part of our environment, and taking into account these interactions, these social epistasis interactions, might help us to better understand things like the speed up of human evolution and like how we've become such a cooperative species. Thank you very much.